The nature of infectious disease, Chapter 14. The learning objectives for this topic are: compare and contrast the terms infection, disease, morbidity, pathogenicity, and virulence. Contrast symptoms, signs, and syndromes. Define etiology. List Koch's postulates, explain their function, and describe their limitations. Explain how microbial extracellular enzymes, toxins, adhesion factors, and anti-phagocytic factors affect virulence. List and describe the 5 typical stages of infectious diseases. The overview for this topic are: manifestations of disease, causation of disease, etiology, virulence factors of infectious agents, stages of infectious diseases. First of our terms describing manifestations of disease, let's expand upon our definition of infection. Infection is when a pathogen has entered the body and has established itself and begun to multiply. But you can be infected and not get a disease, which is any change from health. Most infections are taken care of by your immune system before you feel anything. But anytime you've got pathogens replicating within your body, that's an infection. Disease, as I said, is any change from health. We also call that morbidity. Now, don't get that confused with mortality. Mortality refers to whether you die or not. Let's talk about pathogenicity versus virulence. Pathogenicity refers to the ability of a pathogen to cause disease, and virulence is a measure of how severe that disease is. So these two are not necessarily connected. Rhinovirus is extremely pathogenic. It has a great capacity for causing the common cold in humans, but the common cold is not especially virulent. Very few people die of the common cold. Measles, on the other hand, has a high degree of pathogenicity, has a strong ability to cause disease in humans, and it has a high degree of virulence. In countries where children are not vaccinated against measles, 25% of children under the age of 5 die of measles. That's virulence. Some diseases are more virulent or cause a more severe disease than others. Signs versus symptoms. Signs are those evidences of a change from health or disease that can be measured. So that would be things like a fever. You can put a thermometer in a patient's mouth and you can measure their body temperature and see if they've reached above the normal. Symptoms are those evidences of a disease that are felt only by the patient. So, I'm feeling hot would be a symptom. A sign would be a temperature. I itch all over would be a symptom. A rash would be a sign. Nausea is a symptom. A sign would be vomiting. Hopefully, you'll be able to keep those straight, because I love to ask questions where you have to pick out a symptom from among the signs or vice versa. Now, a syndrome is a collection of symptoms and signs that are typical for a particular disease. And then you have some conditions that are asymptomatic. In other words, the patient doesn't know that they have an infection. Or subclinical conditions. Both of these have a tendency to mean the same thing. So, somebody can have an infection, they don't have a deviation from health, but they can pass that pathogen on to other people who do have disease result. Next up, etiology. Etiology is the study of a cause of disease. Diseases can be caused by hereditary conditions, congenital conditions, or things that you are born with, degenerative conditions. Most of these are age. Nutritional, in other words, if you're malnourished, you can develop a disease. Endocrine, think diabetes. Mental, immunological, think autoimmune diseases. And infectious, in other words, we have a microbe coming in, establishing an infection, and causing a disease. In this class, we're mostly going to be talking about infectious etiology. In any discussion of etiology, we have to go over Koch's postulates. First, a little bit of history into who Koch was. Dr. Robert Koch was a contemporary of Louis Pasteur in the mid-1800s. Dr. Koch was the first person to conclusively tie a microbe to a disease. And in the process of doing that, he established Koch's postulates, which we still use to determine the infectious etiological agent of any disease. First off, the suspected germ, remember the time period he was living in, must be present in every case of the disease. So he was working with anthrax. Anthrax can cause death in mice. So all mice showing symptoms of anthrax, you have to be able to isolate the same gram-positive rod. The healthy subject, if you take blood from the healthy subject, you are not going to find that gram-positive rod. So you have to be able to see it microscopically, and you have to be able to isolate it, which is the next one. The germ must be isolated and grown in pure culture. And Koch developed the process of streaking samples onto agar plates or solid media to get isolated colonies. Next up, the cultured germ must cause the disease when it is inoculated into a healthy, susceptible experimental host, either an animal or plant. So we take that gram-positive rod that we've been able to grow in an isolated colony on agar, inject it into a healthy mouse, and the healthy mouse develops the the signs of anthrax. And the same germ must be re-isolated from the diseased experimental host. Using these postulates, Koch was able to identify the infectious etiological agent of anthrax, tuberculosis, and cholera, just to name a few. Now, there are some diseases where we can't use Koch's postulates. You're probably saying, why not? Well, let's take HIV, AIDS as an example. It would not be ethical to take the HIV virus from a patient that has AIDS and inject it into a healthy human host. Now, if we had animal models, then we could do that, but unfortunately, HIV only infects humans. So we can't use Koch's postulates. We can use most of Koch's postulates. HIV virus is only found in people who have eventually progressed to AIDS. It is not found in healthy populations, with the exception there are some people who are HIV positive who have not progressed. Bacteria and viruses and protozoa always have to throw in the exception. Here's another situation where we can't use Koch's postulates. Periodontal disease. If I take a scraping from the margin of your tooth and your gum, and you've got healthy gums, and I culture that, and I take a scraping from between, the margin of the tooth and gum on a patient that has periodontal disease or inflamed gums, I'm going to find the same organisms. I'm going to find them in different numbers. In other words, when you get a change in the balance between different microbes, then you result in a disease state. But with Koch's postulates, you have to have one organism. So if we can't use Koch's postulates, how do we prove that something is an etiological agent? Well, we do the best we can. We eliminate everything else, like in the example of periodontal disease. We take a look at the numbers and kinds of bacteria that you find in health, compare it to the numbers and kinds of bacteria that you find in periodontal disease, and you arrive at your conclusion of what's causing disease by comparing the two. But you can probably tell it's a whole lot easier to determine the infectious etiological agent when there's just one of them and you only find it in disease, not in health. In other words, when you can apply Koch's postulates. Next, let's talk about virulence factors, or factors that will make a pathogen more virulent or cause more severe disease. So, some organisms, bacteria and protozoa, have extracellular enzymes that allow them to either cause more severe disease or to spread within the host. So, here's a bacterium. It's attached to the epithelial cell. We have a collagen layer, and then we have further inside the dermis. Well, some bacteria can produce an enzyme called hyaluronidase, which breaks up the hyaluronic acid that holds animal cells together. Others, and sometimes the same organism, produce collagenase, which breaks down the collagen, and it allows bacteria to invade deeper into tissues. Some bacteria produce coagulase and kinase. So here's a bacterium that's escaped into the tissues. It can produce a coagulase, which causes a clot to form around it. This hides it from the immune system. Later on, it produces an enzyme called kinase, which breaks down the clot and allows it to escape in to, say, the bloodstream in this situation. Other bacteria produce what are called exotoxins. These are enzymes or proteins that have a bad effect on the host. So in this case, this bacterium secretes a cytotoxin which kills the cell. Most toxins are named after what they attack. So if a toxin attacks the nervous system, it is called a neurotoxin. If a toxin causes gastroenteral distress, it is called an enterotoxin. Now, some bacteria have an endotoxin. The most famous one of all is the endotoxin produced by gram-negative bacteria. It's also known as LPS, lipopolysaccharide. In any case, an endotoxin is a molecule that is a part of the structure of the bacterium, in this case, that is not normally released until the cell dies. Here's a white blood cell. It phagocytizes a gram-negative bacteria, spits out what it doesn't use. Part of this is that LPS, and that causes inflammation, fever, diarrhea, shock, and coagulation of the blood. This also can occur when the gram-negative bacterium dies on its own in the tissues. So you gotta be careful with gram-negative infections. Next up, anti-phagocytic factors. Bacteria that have a capsule are very hard for a phagocyte to phagocytize. It kind of makes them slippery if you want to think of them that way. So the encapsulated bacteria survive to reproduce until antibodies are formed, which makes them easier to phagocytize. We'll get into that in later chapters. Also, incomplete phagocytosis is considered an anti-phagocytic factor. Some pathogens, some of which have a capsule and some of which don't, live just fine in the phagocytic vesicle. They prevent the phagosome from fusing with the lysosome, and they replicate very happily within the white blood cell, and some of them end up eating the white blood cell. So now that we've talked about how pathogens get in, how they cause disease, let's talk about the stages of infectious disease. The first stage is the incubation period. There are no signs or symptoms. Usually you've got a pathogen that gets in, it's causing infection, it's replicating, but the body hasn't noticed yet. Some diseases have what is called the prodromal period. This is vague general symptoms. You just don't feel good. You're starting to sneeze, but you're not sure whether it's a cold or hay fever. Illness is the period of the most severe signs and symptoms. Eventually this will climax. This is when your immune system starts winning, and then you have the decline, where you have declining signs and symptoms as your immune system gets rid of the infectious agent, of the pathogen. And then you have convalescence, wherein you have no signs or symptoms, and your body is repairing any damage that may have happened to the body. Now, be aware that during all of this time you can be infectious, including when you feel just fine. At this point you would be asymptomatic carrier. Here you can be an asymptomatic carrier. Well, that's it for this topic. Once again, reminders of the learning objectives for this topic.